Now we'll move to the next piece of our agenda, which is the Innovation Fast Pitch. In this program, we're going to hear from 4 innovators who are telling their stories and moving the programs that they're working on forward. These handpicked teams were nominated by our High Innovation Program and selected for their stage of development and also how they exemplify different paths for navigating the innovation landscape. These 4 innovators are going to pitch their innovations to you. And so you'll have an opportunity to play our role in selecting and funding a project to move forward. They will each have 10 minutes to present their projects. Chad, here up in the front, will give all the presenters a warning time when the time is almost up. And then this will be followed by live feedback from a panel of expert judges who will be sitting on the far side over there. And then we'll have a Q&A session with all of you in the audience. So we'll have mics to hear your questions clearly, and make sure to raise your hand and don't be shy. Ultimately, remember that pitching is not about perfection. This is going to be about learning faster and getting the feedback you need to move your projects forward than you could alone. Our first, if I could have the judges come up to the front. And while they're doing that, I see there's a number of individuals in the back of the room that are still standing. If there are open seats next to you, if you could raise your hand, and that would be an opportunity for those who are still standing to find a seat for themselves and not have to stand the whole session through. Thank you. So our first judge is Jordan Masanoff. He's the Executive Director of Business Development and Strategy at Amicus Therapeutics. He's a senior biotech leader with more than 15 years of experience in building value for therapeutic programs from preclinical development through commercialization. He brings an end-to-end perspective across business development, portfolio strategy, and commercialization, with a track record that includes multiple M&A licensing transactions and the launch of multiple products in rare disease and specialty markets. He's also had experience in building products and portfolio management functions from the very ground up. Welcome, Jordan. Next, we're pleased to welcome Praful Shah, who's an angel investor focused on social impact, who's invested in more than 75 startup companies. Praful serves on the boards of the Alliance for Collective Action, a nonprofit, and PharmaJet, and his advisor served on the boards of numerous other organizations. Earlier in his career, Praful spent over 30 years as a scientist and R&D executive in pharmaceutical and biotechnology companies. He brings deep technological expertise, investment experience, and a personal passion for alleviating poverty, especially through education and enriching lives through impact-driven innovation. Thanks, Praful. And our third judge is Rhonda Wallen. Rhonda is a dynamic leader with more than 25 years of experience in biotech and medtech industries, serving both as an investor and operator in early and growth stage companies. She brings deep expertise in corporate strategy, go-to-market execution, business development, and market development, with a strong track record of working with partners to increase value and deliver better therapies to patients and physicians faster. Thanks, Rhonda.
Good morning, and thank you for having me here. I'm Allison Dempsey. I'm a psychologist. I'm a professor in our Department of Psychiatry, and most importantly for today, I'm here as the CEO of MindLAMP. We build infrastructure so that healthcare systems can use data in real time to make mental health care more adaptable. In short, we make mental health care dynamic. So I want to start with a story of contrast that I've seen between my personal life as a parent and in my professional life as a psychologist. In my personal life, I have a 12-year-old daughter who most days is amazing, who was diagnosed with type 1 diabetes 2 years ago. We noticed her symptoms the day before Thanksgiving, brought her to Children's Hospital where she received excellent care, and learned quickly about what it's like to help a child whose pancreas does not create or does not function as it should. We learned about the dangers of high blood sugar. We learned about the dangers of low blood sugar and the critical importance of intervening in real time. We were terrified, probably the most terrifying 2 days of my life. And then we went to the Barbara Davis Center that Friday, and we got the best piece of technology that I think is on the market, which is a continuous glucose monitor. It was put on her arm, and she's worn one every single day since then for the last 2 years. This brings us a peace of mind. We're able to look at this regularly to see trends. Just before I came up here, I checked her blood sugar, saw it was starting to trend low, said, hey, you may want to have a Sour Patch Kid, her favorite go-to candy right now. We make small changes daily based on these data, and we make larger changes over time. What you see up here is a graph of her daily data over the course of a week, laid out over time. So we can look at this and say, are there certain times, for example, when she tends to go high? Are there certain times when she tends to go low? Are there certain periods of time, if we look across months, where things seem to be going one way or another? And so we're able to be empowered by this data to improve her care. Now, as a psychologist, things are a little bit different. Instead of getting regular data on a daily or hourly basis, we're getting data maybe every 2 weeks at best. We get data episodically when the patients come into our clinics, and we get a single point of data. Very hard to track trends. It's hard to say what's happening on Tuesdays that's making you so stressed. Oh, you're talking to your mom? That's why you're not sleeping those nights. That's a personal example. And so even though mental health symptoms are just as dynamic, they change daily, sometimes hourly, our care doesn't keep up to that. And that's what MindLAMP exists to fix. Because when we don't have good data, care slows down. We don't adapt treatment as often as it's needed. Treatment takes longer. Patients disengage, and we have longer wait lists, and outcomes suffer. Now, this is a problem for both patients and for providers. Access is limited. 20% of patients who say that they need care can't access it at all. These are data from Colorado. And physicians, especially primary care physicians, where these patients first present most of the time, feel inadequate often to be able to address these symptoms because they don't have the time or the know-how to be able to intervene, and they don't know where to refer because our system has such long wait lists, if they're even open at all. So without timely data, clinicians can't adjust the treatment. Patients feel stuck, they disengage, and care drags on longer than it should. Longer treatment means fewer open slots. Fewer open slots means that less patients are getting care and able to get into care, and the system gets entirely clogged. Now, there is no shortage of mental health technology industry out there. There are a lot of companies. Unfortunately, they don't tend to work to get the patient or help the patient navigate to the right level of care at the right time, and thus ultimately most fail. And this tends to be for one of three reasons. First, they don't integrate into the systems of care where the patients present. The patients have to go and figure out which app is going to be most helpful and download it themselves. Not helpful. The second is that they don't empower patients or the providers with data. Care stays stuck, and they often live just entirely on self-guidance. And third, they live outside of clinical responsibility often. And so what happens is the clinicians aren't involved at all, and they're focused on wellness rather than the treatment of the mental health disorders that the patients are seeking. Patients can't access them. Engagement is low when they can. Outcomes suffer, and systems don't adopt them because they can't scale. But this isn't a tech problem. This is an infrastructure problem. And so we don't need tech solutions, we need infrastructure solutions. So mental health care does not need more single point tools. It doesn't need more wellness apps. It needs infrastructure. It needs to allow care to adapt in real time, and we've built it with MindLAMP. MindLAMP is not another wellness app. It's not a telehealth platform, and it's not a single point tool. Instead, it's an infrastructure designed to live inside systems of care. Now, MindLAMP is built for data-driven care, as I've been saying, with AI in the loop, not humans in the loop, AI in the loop. Humans are driving all the decisions in care. Patients generate data daily. With sensors, we can get things like their activity, their sleep, their social connections. And then we can combine that with daily, what we call ecological momentary assessments. How are you feeling today? How are you feeling in this moment? Are you feeling rested? Are you feeling anxious? What are you stressed about? We can combine all that, synthesize it into those very patterns that I showed you that we have for my daughter's diabetes care. We can look at risk, change, trajectory, and then share this back visibly to the patient and to the provider. So patients can see what's happening. Providers see trajectories. They're not just seeing snapshots, and that's going to improve the decisions that we make in care. Care adapts sooner, patients get better sooner, and we start opening up care to the more patients who need it. Now, this has already been built. We already have this platform. We have a patient app that collects the sensor data, and the patient can put in those ecological momentary assessments. When the data comes back, based on what the clinician programs for the patient, they can also see reminders for these just-in-time care interventions that can be so helpful in the moment when someone's stress has increased or they're having more cravings for substance use, for example. So those data are collected. They go into our database. They then are analyzed by our AI cortex and then visualized for the patient and the clinician so they can make those care decisions together. This is mental health care made dynamic. Low acuity patients get quicker interventions and fewer appointments and with less intensity. And this frees up clinician time to work with the patients who need them more. The more high acuity patients get flagged earlier. They get directed to the right level of treatment at the right time, and the system unsticks. Now, this isn't just theoretical, this is already happening. MindLAMP is out there. It's already impacted thousands of patients. It's in multiple healthcare systems, and it's been incorporated into real clinician workflows. We have generated a lot of data from these 9,000 patients, and this helps us continue to improve the care that we provide and the insights that we're able to get from the data and feed back to those clinicians and to the patients. And we've seen what happens when data is timely. The care adapts faster. The patients stay engaged, outcomes improve, and finally, systems start to move. Now, our data shows this entirely. When care adapts, treatment shortens. Patients start improving in weeks rather than months. Faster improvement means fewer visits, fewer stalled cases, and more capacity for new patients. This is how you unclog the system. Now, we were created within a healthcare system, and we focus on how to incorporate into existing models of care with existing clinicians. We know that healthcare systems don't adopt technology because it's novel. They care about outcomes. They care about making sure that this fits within their regulatory and financial priorities, and they want to make sure that this can improve efficiency of their providers to treat the patients. So we kept all of these in mind as MindLAMP and its care model have been built. Healthcare systems need flow, they need throughput, and they need clinicians focused where it matters. So MindLAMP enables rapid care for low acuity, escalation when risk arises, and efficient use of scarce providers. This is our leadership team. I'm serving as the CEO. I'm a psychologist, as I said, and I also am a clinical informaticist. I represent behavioral health on the physician informatics group for the entire UC Health system. So I know how to improve clinician workflows through technology at a systems level. Dr. John Torres is a psychiatrist. He's also an expert with an international reputation in digital psychiatry, who speaks about privacy, security, and outcomes of these tools. He will serve as our Chief Medical Officer. And Dr. Tiffany Love, who's in the audience, is our Chief Scientific Officer. Dr. Love is a neuroscientist who has deep expertise in remote patient monitoring and sensor data collection. She has worked in the mental health technology industry and specifically knows how to bring these technologies into care redesign in systems. Now, right now, we are academic. We have an academic origin, and that's where we live. But to scale, we need to turn into a company that can support real-world expansion and rapid iteration. And so initial funding would support three things. First of all, we need more nimble coding to improve our data visualization and our user interfaces to get the meaningful data into patients' and providers' hands more quickly in a digestible way that makes it more actionable. Two, we need to improve our EHR and API integrations. Clinicians work in EHR, and so that's where MindLAMP needs to be housed. And finally, we need to start working on initial system-level rollouts with rapid iteration as we go. Mental health care can no longer be reactive. Symptoms change daily, but our infrastructure is not keeping up with it. MindLAMP gives healthcare systems the data and the intelligence to adapt mental health care in real time so clinicians can spend time with the patients who need them most. That's the future we're building. Thank you.
Thanks, Allison. That was a great presentation, and congratulations on the traction. That's amazing. My question has to do with adoption, and I'm curious how, so you have two different constituents, right? You've got your patient has the app on their phone, and then you have the clinicians and the providers who have the dashboard. So that's a good paradigm. I mean, there's a lot of different apps that do that. So how are you getting the patient to adopt this app? Is it driven by their clinician, or are you one of 10,000 apps on the Apple Store?
No. MindLAMP Care is about the infrastructure. And so it's in healthcare or inside healthcare systems, and the clinicians are putting the patients onto the app when the risk is identified so that they can provide that as part of their care paradigm. It is not a direct-to-consumer app.
Question on the MindLAMP. I thought that was an open source platform. Is that correct?
It is right now, and that's what we want to move. Yes.
So was that invented here, or was it already in existence?
The technology was invented by Dr. Torres, who's a member of our team, and the care model and how it integrates into systems was built jointly between CU and between Beth Israel Deaconess Medical Center.
Okay, I have a couple of more comments after Jordan's questions.
Great. Well, yeah, thanks so much. Incredible storytelling. Thanks for sharing all the feedback, particularly as it relates to the customers. I had two questions. One related to the validation of the tool. You mentioned you're an objective, right, to make sure that the data collected is validated and impactful. Could you share a little bit more about how clinicians are actually using the tool, how patients are using the tool? And then I had a question related to how is integration going with some of the customers that you've already identified or that are using your tool?
Yes. So clinicians meet regularly with their patients to go over the data together. And so we will show correlations, for example, between their sensor data and their daily momentary ratings. For example, a patient I worked with, we looked at their data and she looks at it and says, "Wow, I'm really struggling on Mondays." And I said, "Yeah, what do you make of that?" And she says, "I don't like my job," and had not really stopped to think about that. Or like I said, if someone's losing sleep every Wednesday night, I'm wondering what's happening on Wednesday that may be situationally associated with this decrease of sleep. And then can we build a care plan around that? Can we think of just-in-time tools that would be helpful on those nights so that you can disengage from this stressful situation, or can we even change that stressful situation overall? So the data isn't one size fits all. It's used in a care paradigm, and that's why the clinician being at the center of the decision-making with the patient and not the AI is so important. These are data points for them to make sense of with patterns and trajectories, rather than the AI feeding it back.
I'm not sure how much time we have, but anyway, quick. I think for the next presentation, I'm going to have the timer here. You started with a compelling story, which was wonderful. And since I'm supposed to be giving you some feedback on the presentation as an investor, I think it took a long time to get to explain what the problem is and what the solution is. So I think that should have been modified. But overall, the presentation was fantastic for this audience.
Thank you.
Audience questions? Thank you very much for this wonderful presentation. My name is Dana DeBailey. I'm Associate Dean for Research at the Colorado School of Public Health. My question is, and I'm naive to this, my question is whether this infrastructure platform has been tested in a, say, clinical trial, perhaps against standard of care. What is the research showing? Is there a need for that?
Yes. There are multiple clinical trials going on. We actually just finished a 50-person trial here at the university. And so those were the data that we showed actually about even just patient engagement. So whereas for example, in normal clinical trials, we see about 70% of patients remain engaged, we saw 91% in this one. And yeah, there are multiple. Dr. Torres has many going on as well. So, and that's why we have 120 plus publications at this point. Yes.
Yeah. Ivan Strugatsky, angel investor. So there is another important constituency that we need to keep in mind, which are payers. So my question is, based on the experience with the patients that you have, what are the healthcare economics outcomes that you observed, and if those healthcare economics outcomes can be translated and communicated properly to the payers? Thank you.
Yes, great question. And that is future work that we need to do, is a full economic analysis. What I can tell you is that this model supports multiple billing codes, and we've set it up with that in mind. It can be used in the collaborative care models that many primary care clinics are using right now to support mental health. It can be used in traditional outpatient psychotherapy clinics where you bill for some of these services, and can even bill remote patient monitoring codes and or care coordination codes. So much of what we've built, we've made sure fits into systems with that in mind, that we have to bill, we have to be sustainable. And that's what makes us unique compared to a lot of the technology on the market as well.
Allison, hello. So congratulations. You've built up this wonderful asset that is now ready to be unleashed. Yep. But there was no mention of dollars or costs. Mm-hmm. And I'm thinking, maybe it's priceless. But that would be the next step, yeah, right? Is to figure out how to deploy this as a SaaS model for the clinician, mm-hmm, to EMR their way along through. So, I just heard the lead speaker talk about millions and millions of dollars that this organization aggregates and deploys. So are you going to be hiring a CEO who's going to say, with the next $2.5 million, we can do this, that, and the other thing? Because there's a lot of angels in the crowd right here that are looking to back this sort of venture. So the short question is, how much money has been put into the creation of this asset?
Ooh, that's a good question. So first of all, yes, we will have someone more qualified to be a CEO than me when the time comes, because I'm a professor, not an economist or a business person, and we need that knowledge and expertise to lead this to the next level. How much money has come in here into this is a great question. Millions of dollars from funding or from grants, and we need to now move it to payers or to customers paying for it, rather than continuing to try to get grants, which are slower and don't allow us to adapt in real time.
Okay, do we have time for maybe one more question? AJ?
Thank you for the presentation, Allison. AJ Cohen, entrepreneur, investor, all around town commenter. Anyway, you put up a slide regarding the patient's care team, and then you said medical dashboard. And not to insult anybody in the healthcare space here, but how many clinicians or clinics will actually put up a medical dashboard with the ubiquity of wearables, data, and or anecdotal data that can be pushed there and by patient seen as, here's our top 15% of risk of anxiety, depression, suicide, whatever it might be. Now, how will you qualify that? And who are the PAs and MAs that are trained to actually look at that and go, okay, Dr. Dempsey, here's what we're seeing. We need to take action sooner rather than later.
Great question. That's part of our model. We've actually built in what we call digital navigators who are looking at these data regularly, making sure there's good data quality as well, and then feeding this back to the clinician. Now, as a clinician, I can say I'm a convert to this because I have used this now with patients, and I can't imagine trying to do care without this because it gives me such greater insights into care. And it's a single click to get to that. We can feed that into the EHR. So it's a single tab, it's a single click within the workflow to get the data that you need in real time for that patient appointment.
Thank you so much for having me. I'm here to represent RetroNephrix, and our mission is to deliver therapy to the kidney. I'm Heiko Yang, and I am a urologist. I specialize in the surgical removal of kidney stones, and you can think of me as a plumber. I spend a lot of time inside the kidney. It's a really important organ. It filters blood. It excretes the waste in the form of urine. And as a surgeon scientist, I spend a lot of time in the building next door leading a translational research group studying new ways to treat kidney disease. Kidney disease is a huge problem. We all know that. In the US, there's about 37 million adults with some form of chronic kidney disease. That's about 1 in 7. A number of these people will progress to end-stage renal disease, excuse me. And that process is irreversible. Despite all the progress we've made in modern medicine, the best we can do right now is slow down the progression of kidney disease. So why do kidneys fail? Well, kidney failure is caused by damage to nephrons. Nephrons are the functional unit of the kidney. Here's a kidney. Anatomically, it's subdivided into structures called pyramids. And within each pyramid, there are tens of thousands of individual nephrons that span its width. You can think of the nephron as a very long tube that's lined with cells that takes the filtered blood, passes it down its length, and turns it into urine through a number of biochemical processes. We're all born with about 1 million nephrons. Over time, as we get older, get sicker, and acquire conditions that damage the kidney, the cells in the nephrons die, and the number of nephrons that are functioning becomes smaller and smaller. But there are some new therapies that can restore this damage and reverse kidney disease. What are they? Well, there are gene therapies that can reprogram the cells in the nephron to correct mutations and improve their function. CAR T cells can be programmed to remove damaged or cancerous cells. And stem cell therapy promises to replace some of those cells that are lost. But getting those therapies inside the nephron is a huge challenge. It's really, really hard. And this is inherent to how the kidney was designed. In order for any therapeutic to enter the nephron from the bloodstream, so going from here to here, it has to pass through the glomerular filtration barrier. This is a natural barrier that the kidney has to keep the good things in and allow the bad things to come out. It's a filter, and the pores of this filter are very, very small. Each pore is about 8 nanometers. So how small is that? Well, from the perspective of therapeutics, so most small molecules and peptides, they are smaller than this cutoff. They will pass. But for some of these newer therapeutics that I just mentioned, gene vectors, cell therapies, they're flat out too big to pass through. This is a problem that's widely recognized in our field. And there are a lot of really, really smart people working on overcoming this glomerular filtration barrier, using strategies such as lipid engineering, viral modifications, and even using ultrasound and shock waves to overcome this barrier. But unlike those people, I'm not that smart. I don't know how to do any of those things. Like I said in the beginning, I'm more of a plumber. But when I look at this, I can't help but see a plumbing problem. We're trying to get into the plumbing to fix problems, and we're blocked at one end. So why not just go from the other? I call this the RetroNephrix approach to delivering therapeutics. Retro, meaning backwards, nephric, meaning, well, the nephron. We're going backwards up the nephron. And we're going to accomplish this with our invention. It's not a plunger, but it's close. This is an endoscopic needle that's created for the RetroNephrix delivery of therapeutics. I call it the ski pole for all the skiing that we didn't get to do this year. And this is our prototype. What this is going to allow is for urologists, so people just like me, to deliver therapeutics to the kidney using a minimally invasive technique. And this is because our device fits in a ureteroscope, right here. This is a basic tool of the urologist. It allows us to minimally invasively enter the kidney from the bladder and look around. We can identify pyramids. We can identify calyces. Once we've identified a pyramid, we look for the papillary tip right there. And we take our needle, we position it, aim it at the tip, insert it. So this spongy area right here is comprised of structures called the ducts of Bellini. Don't worry, I'm not going to quiz you on this. The ducts of Bellini are the confluence of many, many nephrons upstream. So if you push material into the ducts of Bellini, you can get RetroNephrix distribution. And if you hit one territory and you feel like you missed another, that's okay. You can take the needle out, redirect, find another spot, insert it, and then boom, another RetroNephrix delivery. We've tested this on a human kidney that was donated for research. This is our X-ray of that procedure. You can see our scope here. You can see the needle there. And these wispy black areas, those are 2 RetroNephrix injections that we did. When we take that tissue and we section it, you can see this beautiful RetroNephrix delivery, this distribution. And then histologically, we can see that our tracer material has made it into the tubules of the nephron. This has never been done in a human kidney. So we're really excited about this because of all the possibilities that this can unlock. It's a platform technology for that a lot of pharmaceutical companies and biotech companies will find very helpful because we can treat a lot of things. It's not just one disease. It can be used for any disease involving the nephron, and that's a lot of them, where the therapeutic is either too large to pass through the glomerular filtration barrier, like the ones we just discussed, or the therapy is too toxic, such as chemotherapy, immunosuppression. You can name a number of them. And that's an important point. For the first time, we can deliver a therapy to the kidney that only the kidney sees. So we're excited about this. We're going to refine our prototypes. We're developing that through an engineering team. And what we're really excited about is that we're going to double down on this. And we're using this RetroNephrix approach. We're going to show that we can perform therapeutic gene editing in a human kidney. Our plan is to be the first to CRISPR a human kidney. But back to the needle. We're hoping to have an FDA-approved device by the end of 2028. So currently, our team is small. We're physician-led, but we're well-positioned for success. This is my second startup company. Marshall Stoller, my mentor from UCSF, is a world-renowned urologist and has spun out a lot of successful companies throughout his career. And Kaylin Collins is a close collaborator of mine. She is a gene editing expert. So together, we're developing the needle, and we're also developing our own CRISPR therapeutics. So we're looking for investors, partners, collaborators. What I love most about this work is that what we can achieve is limited only by what we can imagine. And so if this talk has gotten your imagination going and you'd like to work with us, I would love to hear from you. Thank you so much.
Great. Well, thanks for walking us through kind of the narrative, the story there in terms of how the technology works, why it's needed. Just as background, I spent a lot of time thinking about rare renal diseases, and it's a focus area for me. Maybe a piece of feedback and then a question. I would consider just leading with how big the renal space is today. It's booming. It's hot. Renal is back. You probably know it better than us, right? You see the new medications that are coming through, and I think you talked about it at the end, but I think you shortchanged that in terms of the new innovations that are coming that are still have significant unmet need that your technology could solve, which kind of leads to the question, which is, I understand the upside, the value, the promise of gene therapy within the kidney, unproven. But why start there as opposed to looking at all the existing technologies that are essentially not achieving their therapeutic goal that your technology could help support?
Thank you so much for that question. And that's an absolutely valid comment. I mean, there's a million different things that we could try. From a scientific and from a clinical perspective, I think our biggest impact is to be able to do something, not make an incremental change, but to make an order of magnitude change. And for us, that space lives in CRISPR therapeutics and being able to durably edit the kidney, correct mutations, and cure disease. That is one of the biggest unmet needs in kidney disease, and we're hoping to achieve that using this.
Thanks for that answer, because it leads nicely into my question. So the patient population that you're looking at for this CRISPR proof of concept, because that was also one of my questions, why start with gene therapy, CRISPR in particular, but are you looking to reverse end-stage renal disease? And so you're enrolling people who have already failed, or like, who are you thinking is your ideal patient for this?
So thank you for that question. I think if we work backwards from what causes end-stage renal disease, what are the monogenic causes of that? The frequent flyers that always come up, polycystic kidney disease, right? That is something that is inevitable. Almost for a lot of patients, they live their life and they wait to get on dialysis and get a transplant. That is caused by a single gene mutation. And that is something where there are a lot of different CRISPR tools being developed to correct that mutation, but no way to deliver it. And so if we start with that as a cause of end-stage renal disease, I think that is a really great use case for our technology.
So just to clarify, or maybe you can educate me on the sub-markets or the different clinical cases of kidney disease, but my understanding was a lot of ESRD is from diabetes, right? What's the relative patient population there?
Oh, yeah. So totally, there's a number of different causes of end-stage renal disease, all different mechanisms. We have to start somewhere. So we're going to start with the monogenic diseases, but then we're going to work our way towards things like diabetes, hypertension, all of these different things that aren't even thought of as genetic diseases, but could stand to benefit from a single gene target that we can correct.
That's great. So just for your investor type presentations, maybe just talk a little bit about like this polycystic, single gene mutation, because I think a lot of people think kidney disease and they think diabetes.
Yeah.
And that's not who you're going after.
Not yet, at least.
Right. Okay.
Yeah.
Thank you.
Yep.
So following up on that, who is your patient population and how would you go about getting this device to them?
Yeah. So our patient population, like I said, is myriad. We have so many different indications, but let's start with one. Let's start with polycystic kidney disease. So for these patients, we are going to partner with a pharmaceutical company that already has either an AAV or lipid nanoparticle-based therapeutic. We're going to develop our delivery method. We're going to prove that it works. We're going to prove that it's safe. We're going to get it FDA approved. And then we're going to teach urologists how to do this procedure. And our polycystic kidney disease patients are going to be the first patients in our clinical trial.
From an investor perspective, how much money have you raised? How much money do you need? How much when do you need it?
Great question. We are brand spanking new. So to answer that question, to meet our milestones, at least for things like prototype development, preclinical testing, the amount of fundraising we have to do is actually quite minimal. We're actually going to use a lot of research money through research funding to develop that data. But where we need help is in the FDA approval, the regulatory approval process. And so that means having regulatory folks, people to interface with the FDA, and then also people like having a CEO. That would be very important to us.
I would be amazed if I didn't mention that HeikuYangLab.com, which is your website, has a lot of really useful information.
Oh, thank you. Yes, that is my website, HeikuYangLab.com. If you'd like to visit it. We have, don't do it before lunch. Maybe wait until after.
Any audience questions? We also have a mic in the back.
Hi, Ivan Stugatsky, angel investor. So, two-part question. Number one, kind of, what do you think would be the reimbursement paths for you? Because you have separately the device and separately the treatment. And then the second question, I noticed that you are pursuing 510K pathway. What is the predicate device?
Let me answer the second question first. So the 510K pathway says that there is something at least reasonably similar that has already been FDA approved. There is something that's already out there for injection in urologic structures, and it's called the Williams needle. It's used to inject Botox into the bladder, and that's an endoscopic needle as well. So that's hopefully the device that we're going to use as a predicate. I think there's also something for the brain that I recently learned about, where you can deploy little stents in the brain that has a little catheter and a needle. But anyway, before that, your question was about the reimbursement for, you mean for the company or for the device itself? Yeah. So the easier easier question to answer is for clinicians. So with any new procedure, you want to get a CPT code for a new procedure, and you need to advocate for what that value is and justify why it's being used. So that's number one. And I think at least how I envision this is that it should be something that is very worth urologists' time. Now, in terms of how we plan as a business strategy to partner with other companies, it's going to be going in sub-licensing for different indications and partnering for drug device combinations for their therapeutics. Yes.
Dr. Yang, thank you so much for taking us through the kidney like you did. So you started out with kidney stones and you wound up with CRISPR gene editing. I'm questioning, is there a gene that's causing kidney stones? And then do you have to have your needle to deliver the gene therapy? Is that that's the bottleneck right there?
Yeah. So I'll answer that one first. So that's another 10 slides that I deleted. There are other folks out there trying this RetroNephrix delivery. A lot of folks using pressurization, retroureteral injection, things like that. It doesn't work. It does not work in the human kidney. And no one knows that yet because no one has access to human kidneys the way we have a partnership with Donor Alliance Donor Network West, and all these organ procurement organizations where we get human kidneys that are donated for research. So I'm letting you in on this secret because I trust you all, and I trust you will not tell all the other groups working on this. But that is the key, is that this is the bottleneck, and this is the only way that we can do it. Sorry, your original question, or first question was?
Well, you also said you're brand spanking new, and it looks to me like you have put a lot of money and time and energy in this. It's probably a lot of grant monies. Is that fair?
Yeah, yes, sir. So how did I start with the kidney stones? Well, again, I think this illustrates why this is something that our group, why I was able to come up with this idea, is that you can only understand how to do this if you're a urologist. Most urologists don't spend time thinking about delivery or CRISPR therapeutics or anything like that. So as a surgeon scientist, where I understand what the need is for the next generation of kidney therapies, this is something I can identify, and I have the skill set and the tools to be able to execute something like this. I bet you no one else probably has thought about doing it this way, because no one else is a urologist that knows that you can do it this way.
Let's give a round of applause to Dr. Heiko.
Thank you.
Today. My name is Tracy Bale. I am a professor in the Department of Psychiatry and a neuroscientist here at CU Anschutz. And I am the Chief Scientific Officer and co-founder of our brand new company, NeuroVesica, the biomarker platform for precision psychiatry. And NeuroVesica is going to make psychiatry much more precise, guiding us into better diagnoses and better treatment. So psychiatrists are not failing us. Science has failed psychiatry. Currently, our tools just don't work. 20% of Americans are currently suffering with a diagnosable mental illness. That's 65 million Americans, over 1 billion people globally. This costs American taxpayers over $477 billion annually. The problem are manyfold. I'm going to start off with one: diagnosis. Diagnosis in psychiatry, as you heard from Allison earlier, and the way in which we diagnose individuals with a mental illness is subjective. Why is that a problem? Let me give you an example. You have cancer. You're sitting in your doctor's office. She walks in and sits down and she says, "How does your tumor feel today?" That doesn't happen in any other medical field except psychiatry. No biomarkers, no blood tests, no imaging. In psychiatry, it is your report of how you are feeling and your physician's subjective interpretation of that information. We need to do better. We need objective biomarkers to lessen the suffering in mental illness. Treatment is also a problem. Currently, treatment is a lot of guesswork by your physician. Many of you in this audience are family members, friends, family, have experienced the issues of 2, 3, 4, 5 or more medications, changing the dose, and still the suffering continues over the course of years. Psychiatrists are not failing. Science has failed psychiatry. We need better objective markers. And lastly, one of the big issues in psychiatry is our lack of really good drug development. We have some new drugs out there, but the decrease in the clinical trials has been a problem. And in fact, in the last 25 years, 90% of clinical trials in psychiatry have failed. And the result of that is that drug companies starting new drug clinical trials has also decreased by 90% in the last decade, leaving us without novel indications and novel drugs of which we go after. So why do clinical trials fail? This is something that NeuroVesica is prepared to address immediately. Currently, clinical trials fail in part because of things like the placebo effect. These graphs here are an example of that. I'm showing you clinical trials in Site A and Site B, the same drug being used to treat treatment-resistant depression. You can see by a decrease in the slope there for drug treatment, which is improving mood, but the difference between the two sites is not drug, it's placebo. And the problem is in clinical trials, just like in the clinic, that we have subjective measures that are utilized to test for improvement. We have no objective measures. Without objective measures, we cannot distinguish between placebo effect and drug effect, and therefore clinical trials fail. And in fact, 50% of those clinical trials in psychiatry have failed because they cannot beat placebo. So another example I want to give you is in addition of why clinical trials fail in psychiatry has a lot to do with the fact that in psychiatry, we look at subjective measures, and those can be determined by the drug companies initiating the clinical trial, and they can select the subjective measures that they want to use, and they can differ. What I'm showing you here are two very similar drugs, clinical trial A and clinical trial B. You can see the drug effect and placebo effect are very similar in both. Now, if I were to ask you as an audience, which one do you think succeeded as a clinical trial? So see a show of hands who think it's A. Okay, let's see a show of hands who thinks it's B. Okay, and in fact, it was clinical trial A that succeeded, but they look very similar. The difference between them was really the subjective measures that the drug companies selected to utilize as their predetermined outcomes. When you go into a clinical trial, you have to say what your outcomes are going to be. Clinical trial B selected a different one than A, and they failed. And not only was their clinical trial considered a failure, but that drug company also failed because of that clinical trial, because trials are very expensive. So we not only lost a drug opportunity, we lost another company in this space, and that's very valuable. Subjective measures are very problematic across psychiatry. They can be inconsistent, they're often unreliable, resulting in clinical trial failure. NeuroVesica is prepared to address this. We focus on extracellular vesicles, or EVs, as real-time reporters. EVs are messengers that can tell us what is working and what is not. So what are they? Well, EVs are nanoparticles that are traveling in very high concentration in your bloodstream, making them easily accessible, easy for us to access with a blood draw. They are the body's communication. These EVs allow us to ask in cell-to-cell and tissue-to-tissue communication. We can learn a lot about your biology with one blood sample. These EVs carry a lot of proteins that are coming from the cells that they are being delivered from and acting on different cells, such in this case I'm showing you here is the immune system that allows us, NeuroVesica, can utilize proteomics assessment to build out our platform to be able to determine exactly these signals, to know who is responding to treatment or who has what disease. EVs are also dynamically changing all the time, but they very much determine based on the state of your disease and the response to your drug treatment, allowing us to assess treatment responders and placebo responders and distinguish between them as predictive models of objective markers. The key here that NeuroVesica has, we are one of a kind, revolutionizing how we think about psychiatry, is a biomarker prediction response. We are the only company modeling EVs and dynamic response with longitudinal studies in treatment response. Other companies, including in other fields such as cancer, focus a lot on things like static time points, RNA levels in these EVs, single time points assessments, and group level averages. Why is that a problem? Because groups don't come into a clinical trial and ask to enroll. Groups don't walk into your doctor's office. Individuals do. We need to be able to assess objectively the individual. NeuroVesica uses dynamic proteomic changes. From start to finish, we can predict who will respond to a treatment and who will have a durable response, the outcomes we're very much looking for in psychiatry. We use repeated sampling across longitudinal data sets. That way, we can utilize this data at the individual level for the first time in psychiatry to predict who will be a treatment responder appropriate for a given clinical trial, who in a doctor's office will be able to seek treatment, and who will be a non-responder or a placebo responder. Our platform is built with an end-to-end engine. We can start with blood sampling, isolate EVs, perform our proteomics by mass spectrometry. Using machine learning and AI, we are able to build a large platform that encompasses across transdiagnosis in psychiatry, building out a predictive signature for the first time. This is scalable, longitudinal, multimodal, and clinically integratable. NeuroVesica is the first company in this space to build out a comprehensive transdiagnostic platform. It is broadly longitudinal and treatment responsive, allowing EV data sets across psychiatric diagnosis, inclusive of affective disorders, neurodevelopmental disorders, addiction, even trauma exposure across the lifespan, and most importantly, also aging, including diseases of age, including neurodegeneration. Our team is small but mighty. We have expertise in neuroscience and psychiatry across EV biology. As the chief scientific officer and neuroscientist and co-founder of this company, I have expertise in neuroscience. I also have expertise in EV biology for preclinical and clinical models. Dr. Neill Epperson is a psychiatrist. She is the chief medical officer and co-founder of NeuroVesica, and she has over 30 years experience in this space of psychiatry. Dr. Rodrigo Orso is our director of research. He has the research expertise for study design and mechanisms really critical for EV isolation, validation, and how we do our analyses important for building out proteomic models. Arthur Feltrin is our director of science and analytics, really critical in this space to be able to take the data and pump it for everything it's worth, building out these predictive models across disease and treatment states. Okay, so our business strategy has a three-stage approach. First is research services. How many biotech companies begin and have no way to generate revenue? Most of them. NeuroVesica has the ability from day one to develop through research services, partnering with biotech and pharmaceutical companies, making their clinical trials more successful. We can do this by giving them objective outcomes, reducing their placebo effect, and excluding all of their treatment non-responders. Utilizing that revenue, we then begin our assessment for clinical diagnostics. As you heard earlier from Allison, this is a huge problem in the diagnosis space, and being able to provide objective markers in the clinic moves people through more accurately and more quickly. Obviously, this requires a lot of partnering, not only with psychiatry clinics, but also with health insurance companies, allowing repayment for those blood tests that we develop, giving them, again, objective measures for diagnosis and treatment. Then moving into our third stage, we will be able to use our proteomics platform to identify very novel therapeutic targets, thinking outside the box instead of always inside the box of the brain. Our evaluation is based on several different things, including our research services. As I told you earlier, for drug companies to launch into a phase 3 trial costs anywhere from $200 million to $1 billion per drug. So saving them all of that revenue is a really good investment. In the diagnostics market, expected to exceed $20 billion in the coming years, and we already know that the antidepressant drug market is booming right now, expectations of more than $30 billion in revenue. NeuroVesica timeline is showing you here. We were the first to be able to launch into securing proprietary samples that help us build this platform, including last summer where we entered into access to over 2,000 blood samples in a 17-year mom and baby longitudinal study. We also finalized a deal for additional multimodality data set where they've assessed individuals as they progress through aging from ages 50 all the way through the time that they've passed away, and those repeated data sets, allowing us to assess changes in these EV biomarkers with aging. This spring, we just finalized our treatment-resistant depression clinical trial for ketamine, and by this summer, we'll have a combination of addiction for GLP-1 treatment of alcohol use disorder, psilocybin treatment and treatment-resistant depression, and comparisons of ketamine and S-ketamine samples, and also in treatment-resistant depression. So where are we going with this? Building out this giant platform that NeuroVesica has invested in allows us to develop a NeuroVesica index. Utilizing that index into our ability for a proprietary assessment of stratifying subjects prior to clinical trials entrance, we can save pharmaceutical companies money, time, leading to better development of drugs and faster. And I'm going to show you an example right here of our brand new data set where NeuroVesica was able to, in completion of a clinical trial related to ketamine treatment for treatment-resistant depression, you can see here on this graph a response rate, meaning of all the subjects in this clinical trial, What percent of them showed a therapeutic response to the drug ketamine? And as you can see across the x-axis, the severity of depression. Even in the most severe cases of depression with treatment by ketamine, prior to pre-stratification with the NeuroVesica index, you can maybe get just around 50% of your subjects responding. Once we apply the NeuroVesica index to this data set and reevaluate response, we can now get them up to over 90% response. That would save millions, if not $1 billion, for those pharmaceutical companies. So NeuroVesica is making psychiatry precise. Using our proprietary platform, we can improve clinical trial success right out of the gate. These are shovel-ready platform. Accelerating time to market, inclusion optimization, and preventing, most importantly, clinical trial failure. By stratifying subjects who will be appropriate for this trial and who will not, unlocking precision psychiatry for improved diagnosis and treatment, and ultimately, and most importantly, changing lives for millions. NeuroVesica, again, the biomarker platform for precision psychiatry, the first of its kind. I want to thank you for your attention, and I really want to give a shout out to CU Innovations for their incredible work in putting this all together, and my contact information there, and I'm happy to take questions.
Great presentations. It's interesting, your talk came right after Dr. Wang's, Yang's, I mean, and it just so happens that vesica means bladder.
Also means vesicle, but okay.
You're right, yes. This is so interesting. I spent a lot of time in drug development, and part of the reasons we never did come up with the right drug is we never could identify correct diagnosis, or it's so subjective, couldn't actually get to that. The question I have is, do you have an existing drug where you can actually say, okay, by the way, we actually went back and just said, took, Prozac or something and give it to the patients and see how your platform works out against the numbers you have?
So redoing a clinical trial, that would be incredibly expensive, but what we're currently working on is partnerships to test and evaluate our models to assess from those platforms, using our platform with drug companies who have already done trials and have blood samples stored.
Thank you.
Yeah. Great presentation. Yeah, it's amazing, right? This is an example of a space where there's been so much innovation, yet so much unmet need, and the status quo kind of leads the way here. Kind of leads to have two questions. One is, just on the technology side, could you share a little bit more about connect the dots between the EVs and the predictive signature? Exactly like what is in there that makes you confident that you're going to be able to help identify better patients for clinical trials? And then second is, as with your customer discovery and pharma companies, is the expectation that they're going to lead the way in overcoming some of the inertia with FDA and regulators who have, standard endpoints and standard trials, the reasons that companies continue to kind of fall into the same trap of studying products like this in the same way? Or is the expectation that you're going to lead the way and kind of change the paradigm for FDA as it relates to biomarkers?
Okay, so for the first question, what's in our proprietary platform? It's like asking Colonel Sanders, what's in your recipe?
Just maybe you call it a biomarker approach, and then you have some...
The biomarker approach, so using the isolation of the extracellular vesicles, performing mass spectrometry on the protein, so it's a protein-based platform. Not much more I'm not going to tell you the specific proteins because that of course would be giving away the whole show, but it's protein based of what is in a given individual's, either initially, we can tell from your blood sample initially whether you will be a treatment responder, or if you're looking for a durable outcome, so more than just acute 24 hours, I feel better, but then you're back in a week, durable outcome.
There's more around just the validation of these biomarkers. Something in your proteomics shows up and you're connecting that to some form of CNS disorder or disease. And have you validated that in a way that drug companies.
Okay, so I see where you're going with this. So the beautiful thing about extracellular vesicles, or if you want to classify as a biomarker, biomarkers can mean a lot of things to a lot of people. And a lot of biomarkers have been tried in psychiatry, and none of them have made it to clinic. We have currently zero biomarkers in psychiatry. Nothing has moved that far. A biomarker can be something that's just predictive of your current state and biologically itself tell you nothing. Or if you're really lucky, the biomarker can tell you also something functionally relevant because of it's working with the immune system, et cetera, something like that, or some interaction with the brain. So in our platform, we have a mixture of all of that, depending on what state a drug company would be looking at. Again, are they looking for an acute validation, or are they looking for a durable outcome?
And sorry, just the other question was, are you planning on leading the way with FDA to kind of change the paradigm in terms of how they think about clinical trials, or is the expectation that you're
This is a really important question. I think for the initial research services part of it, I don't think it has to involve the FDA because it's not an outcome. This is a stratification for inclusion-exclusion criteria that would enroll people into the next steps that we'd be working on for the diagnostics and the drug development. Those, of course, are going to be more dependent on FDA.
Thanks, Tracy. Really good description of the problem. And yeah, biomarkers in psychiatry are really hard. As far as the solution and really understanding the amount of work that goes into developing a proteomic classifier, which is what you're doing, that's, I think, a bit understated here. So one of the things I wanted to get a little bit more from you on is training versus validation. I think one of the reasons we haven't seen a lot of biomarkers come through is because FDA, to Jordan's point, wants some prospective validation. And all of these are developed on retrospective analysis of older data sets. So that's one issue I'm trying to get your thoughts on. And then, yeah, just in terms of, again, those data sets that you're doing your training on, what then are you doing your validation on?
Yep. So I think that was a question earlier, which is the question is related to training your model, right? How do you develop a reliable model, which is what we've been doing for the past several years, and doing expansive assessments across different neuropsychiatric diseases. And so building that model together and as part of the training is what we've done thus far. And then doing the partnerships with different drug companies that already have run well-designed clinical trials that we can utilize their blood samples to then blindedly look at how our model works for those studies and the predictive outcomes.
That's great, because getting those data sets is super hard for the first part. So I like that strategy. Thank you.
Thank you.
To parlay is to have a conversation. But I bet you heard that word first in a pirate movie. And that's because for pirates, parlay didn't mean just talking at each other. No. This was a sacred vow to really hear what the other person had to say. Unfortunately, there are millions of people for whom it is impossible to make that vow. And this is a horrifying fact because we know that 1 in 12 cases of dementia is due to untreated hearing loss. What's more, if you find yourself struggling to hear in these networking events, well, you're just like 60 million other Americans, and unfortunately, your lifetime risk of dementia is twice that. Why? Simply put, the brain craves activity. This can come from a direct input like your sense of hearing, but the reason hearing loss is so profound, people choose to withdraw from their social and their professional lives, and that deprives the brain of even more activity and ultimately kicks off a cascade that ends in that cognitive decline. What you may not know, though, there's actually 2 forms of hearing loss. There's the familiar damage to one's ear. Think cochleas or hair cells, maybe those little bones. But this is what we call peripheral hearing loss, and is by and large a volume problem. That means we can fix it with hearing aids because they amplify all those sounds around you. This is not going to work if you can't hear in noisy places. It's actually going to make the problem worse more often than not. And that's because those 60 million Americans who struggle to hear in noisy places struggle with a processing deficit, what we call central hearing loss. We set out to solve this problem. But to do so, we had to understand, how does one hear in a noisy place? And it turns out you have in your brainstem specifically, a very special circuit. It figures out where in space around you different sounds come from. It does so by being the literal fastest circuit in your brain. Now, the way you make one circuit faster than another, you can wrap the nerves in something called myelin. And the thicker that myelin wraps around a nerve, the faster it can communicate. Conversely, and herein lies the problem, thinner myelin means slower communication. And for those 60 million Americans, well, the myelin in their brainstem has deteriorated, causing that circuit to slow down. But this suggests a way forward, doesn't it? And that's our breakthrough, what we call localized oligodendrocyte optimization therapy, or if you like our pirate theme, LOOT. And LOOT is a first of its kind treatment for this neurologic root cause. It is a 30-day treatment that consists of an hour a day of listening to an audio therapy, combined with a drug called clemistine fumarate that's been long approved by the FDA as an antihistamine. But I'd like to emphasize, this is a combination, and it only works if you do both things. This is because that drug clemistine, at the dose we prescribe, is no longer just an antihistamine. It now also drives the cells that make myelin to be able to do so, those oligodendrocytes. But the oligodendrocytes don't know where to deposit that myelin without high levels of neural circuit activity. And that is what our 30-day audio therapy is doing. We will be distributing LOOT through proven business paths. And I'm going to wager that everybody in this room knows somebody or has personally paid out of pocket for life-altering therapy. Maybe it was LASIK or GLP-1s, or yes, even hearing aids. And in the case of hearing aids specifically, patients will pay on average $5,000 to their audiologist or ENT. That clinician, by the way, had purchased that device from the manufacturer for about $2,500. Now, this is a really useful distribution model because audiologists and ENTs are the most equipped healthcare providers to identify peripheral or central hearing loss, which, by the way, patients can have both of. Now, a kind of crazy statistic, just to set how big this market really is, it turns out that every hour, 5,000 Americans are going to go to their audiologist, and central hearing loss will be their diagnosis. To say that differently, since we all entered this room to when I'm talking, there have been roughly 10,000 people that went to their care provider seeking help and were sent home with nothing because there is no treatment today. But that's where we come in. Parley is offering a complete solution, and we do so at a price competitive with hearing aids, so we know that the market will tolerate it. Our solution comes in the form of a therapy portal that allows us to stream in a DRM-protected manner the patient's daily therapy, right in the comfort of their own home. We have baked into this portal a lot of very important neurologic data capture, in addition to the specialized hardware that we provide the patients, a set of headphones, which we've partnered with Jabra to do so, that allow us to monitor the patient's compliance during their at-home therapy. This is essential for audiologists to actually monitor and ensure the patients comply. Now, an important point about audiologists, they can't actually write prescriptions. So even though they are the most appropriate clinician to diagnose the problem, they do have a bit of a roadblock. So we offer the solution there as well with our telemedicine capability. Now, I mentioned DRM very briefly. If you're not familiar, this is digital rights media, and it's literally the legal framework that allows every streaming platform you've ever used to operate. But that's not where our defensibility ends. We've already submitted 2 provisional patents. These will convert into 5 total patents that cover a lot of our other innovations, including the neural data that we will be capturing and our strategies to mine this data. I'm Sam Budoff. I'm very proud to be the captain of this ship, and with my co-founder, Dr. Achim Klug, we are going to solve this problem. Listen, Achim and I are both PhD neuroscientists, so we know a thing or two about brains. We also have a combined 20 years of experience building biotech and neurotech startups. So we know how to steer the ship. But the real reason we're going to succeed where no one else could is because Achim has dedicated the last 30 years of his life, ever since he looked like that, to really understanding at a neural level how hearing and noise works. And he's done so, as Kim told us, at this incredible institution. And that's important because the last 7 years of his research efforts have been dedicated to this translational problem. He was awarded $7 million by the NIH to fund his demyelination hypothesis, which he proved in neurosurgical animal experiments. These led as well to the preclinical data that underlies our company formation, the patents we've submitted, 2 additional R01 awards his lab just earned for $6 million. And most importantly, the data the FDA agreed meant it was imperative we start our clinical study. And on that point, I'm really excited to tell you, we haven't raised a dilutive dollar yet, but we are today, and that is already as a phase 2 clinical stage company. So we are raising for our already active and enrolling study right here at Anschutz. And if you would like to participate, we encourage you to volunteer. And if you do so, you're going to find yourself in this sound chamber. It's a really cool little piece of equipment. And just like Tracy was telling us, it is imperative to have a quantitative endpoint when you are running any kind of neurologic study. And that's what this does. This allows us to measure in an unambiguous way how much improvement we provide for patients. But we're also running a clinical study. So safety is something that most companies worry about. We don't. And that's not because we don't care. Far from it. It's because this exact drug at this exact dosage has already cleared phase 2. So we know it's safe. And because of that, we have the conviction to take advantage of a brand new regulatory framework that allows us to sell our experimental treatment this year in the state of Montana. We already have our first customer, an audiologist who will be distributing this. And even if we don't get any more, our worst case pro formas still show us breaking even before FDA market approval. But the reason to be excited about this is not that market test which will let us establish price point, but the fact that we will have established price point. This gives us provider buy-in for when it comes time to get a CPT code, or, and most importantly, the price point data that the insurers need to offer us reimbursement under a HCPCS code. Now, when we do get that FDA approval, we will be distributing through the 5 major equipment providers in the US. They service the 20,000 roughly audiologists and ENTs here. This allows us to tap into an annual $9 billion market. The way we've engineered our platform, though, further down the line, we will be able to expand to direct-to-consumer in a much, much larger market. But bringing it back to today, we're seeking $3 million right now. This will let us complete that already enrolling phase 2 study. It'll also let us kick off that market test. But if you don't have $3 billion today, $3 million, you can vote for us. That'll still help us fund this trial. I'd like to leave you with this thought, though. Our scale is not limited like hearing aids. Consider the fact that most adults think they should take baby aspirin to prevent a heart attack. The reality is that is only going to prevent 1 in 2,500 heart attacks. On the other hand, treating hearing loss will prevent 1 in 12 cases of dementia. And if you find yourself struggling today after I kind of got this into your head, well, you're at twice the risk. So it's very important that we fix that. So if you'd like to join our crew, sorry, let's parlay.
Interesting presentation. I think I heard most of it. I'm an old man.
You might want to enroll in our study then.
You talked about myelination. That kind of got my curiosity. Is there any correlation between hearing loss, the cocktail party syndrome, and people who have MS?
So that's a really interesting question. I can't tell you the answer to that today. But what I can tell you is that the multiple sclerosis studies are the ones that led us to identifying this particular drug and why we have so much conviction it's going to work. And I'll mention as well, those MS studies, they focused on a biomarker on the optic nerve. And those studies show success in humans, that is effective translation on that nerve. But that's a nerve that's very highly stimulated. So just like our preclinical work, we know that you need the combination of high levels of neural circuit activation with the drug to see a meaningful effect.
Changing the subject to investor, you're raising $3 million.
Yep.
I assume you're going to do something with it?
Oh, yeah. So that's going to fund that clinical trial to completion, and it's going to let us kick off that market test.
What are the terms? Is $3 million, does it give me 50% of the company or 5% of the company?
We can talk offline, but I would say closer to 5.
Okay, thank you.
Great presentation, Sam.
Thank you.
So I'm very familiar with this, so I'm going to drill down on one little nitpicky point that I didn't quite understand, and that's on the market. I think you said it was a $9 billion market, and then you had sort of the parenthetical how you got to that. And there was something there that said every 5 years. So are you thinking that a patient will need to refresh either the headphones or the treatments every 5 years?
It's a really great question. So this is not something we're trying to prove in this particular clinical trial, but when we look at the literature about how oligodendrocytes work in the human brain, we know that they hang out for about 5 years. Now, this is as opposed to neurons. You're born with pretty much all the ones you're going to die with. Little asterisk on that statement, but oligodendrocytes, they need to renew every 5 years or so. So for that reason, we suspect what is actually causing this problem in at least older individuals is those oligodendrocytes are not properly turning over. So our treatment is making them do just that. Now, this implies that every 5 years, a patient would need to repeat that 30-day therapy. But asking somebody to do something for a month every 5 years, we think that this is something that most people will tolerate.
Yeah, great presentation, really compelling. Appreciate the business model and the pathway and the market, like very clear there. I may have missed it though. I know you're going into phase 2 studies, but is there any data today that gives confidence that the combination will work or that the sound therapy in and of itself is going to be effective in helping with hearing loss and dementia and other things like that? It just felt like it was missing some just kind of core data.
No, absolutely. So, we've done a lot of investor pitches, as you can probably guess, and turns out investors don't usually want to see the science data, which is a difficult thing for Achim and I. But the preclinical data is very, very clear. So we have 4 conditions in that preclinical study, and we've replicated that for the phase 2 study. These conditions are sound plus drug, a placebo sound plus the real drug, a placebo drug plus the real sound, and then placebo. So in those animal studies, it is only in that combination case with the real active sound and drug that we see an effect. And this mirrors the multiple sclerosis study I just mentioned, where the optic nerve is remyelinated. But if you look at other studies of clemistine that are looking at more general biomarkers, say like a CSF proteomic sample, they actually don't reveal a significant improvement because that effect is localized to circuits.
Sam, I'm so glad I just saw this presentation. I have some hearing problems myself, and I want to be part of your experiments.
Please do.
There's a big angel festival going on at the Westminster. There's a couple of hundred angels over there that probably want to meet you to talk about that 3 million seed round. How much money is put into the company so far, and what are the animal models that are working here? Because I don't see the mice raising their hands, say, I heard the tone.
No, it's a great point. And so a couple answers here. So to date, it's all been non-dilutive funding. So there was $7 million initially invested by the NIH that funded that demyelination hypothesis and all of our preclinical work. Since then, CU Innovations has been incredibly helpful. We have a Spark Award. And we just got $6 million more to Achim's lab for basically IND supporting basic research. Now, this cannot be used for animal studies, but it still is a very important part of the package. As far as those animals, so we use Mongolian gerbils. Now, this is not for any fun reason. It's because Mongolian gerbils are actually an almost perfect analog to the human auditory system in the form of a rodent. So mice and rats, they hear an ultrasound. That means their brains are specialized for hearing an ultrasound. So it's not a great model, even though we do have really cool genetic tools, and there's a whole interesting story on that. But the gerbil, because it has basically the same auditory brain function as humans, all you have to do to give it this disorder is basically let them hang out for 2 years, all you can eat, buffet, that kind of good stuff. And they will naturally age into having this phenotype. So a 2-year-old gerbil is basically the equivalent of a 65-year-old person. Now here's a bonus. Most people, they live their life, they go to rock concerts, they stand near a jet maybe, and they will actually damage that cochlea. They will induce some form of peripheral hearing loss potentially. Gerbils hanging out in that all-you-can-eat buffet, no chance of them getting peripheral hearing damage. They only have the central phenotype. And now this is actually an important part of our clinical study. We replicate this by screening out individuals with the peripheral hearing loss so that we have a pure cohort to really prove efficacy as cleanly as possible.
Last question.
Okay. Thank you, Sam. I loved your presentation because you're one of the few from previous presenters that you shared with us on your intellectual property. And I'm a Angel Capital Association member, as well as a member for Curious Forum. But my background is mechanical and electronics engineering. So I'm going to ask some very ignorant question. So first of all, what exactly is it? Because you talked about the sound, you talked about, you know, and the earphone, and also drugs. So therefore, you know, is that some kind of just a therapy, or is it a combination of something? That's the first question. The second question, because of this, so is this going, what kind of regulatory pathway do you have to go through from FDA? Thank you.
Thank you. Great questions. So I'll emphasize we have 2 provisional patents that have been submitted. These will convert into 5 total patents. So the first patent, arguably the most important, is the one we licensed from CU Innovations. And this is the patent that covers the combination therapy, that idea of sound stimulation plus drug. And I'll mention we have about 40 drugs listed in there, even though our trial is only focused on one. And this basically gives us a very broad protection to operate as really the only company that can use this kind of lute style therapy. Now, as far as our second provisional, we filed that as the company itself. And this patent covers a lot of other important ideas, including a lot of hardware and software innovations to bolster the way we deliver this. So just as one example, we have baked into the audio files that we stream to patients, digital watermarks, sonic watermarks. So the patient isn't going to notice this. It's going to sound like just maybe some kind of beeping or something. You know, it'll seem like one of the other stimuli. And this is important because if there's a bad actor who is trying to rip us off or even just put it on the internet, we will know exactly who it was because their IP address, when they did this, et cetera. That is all baked into the file they would have just stolen. And, you know, as I mentioned, DRM protection is what is really going to allow us to take down anything that is trying to do this. And it's going to protect our business the same way Netflix, Apple, you know, pick your favorite streaming platform. All of those companies rely on DRM to protect these kind of digital assets.
All right. Well, before we go to Vindel's talk, I do want to announce the Innovation Forum Audience Favorite Award. So we had a discussion in the back room, and actually this was a tie between 2 different teams. So what we'll be doing is we'll be splitting the award between NeuroVesica and Parley Neurotech. So congratulations to the teams.